show the yellowish green can be count the number under the fluorescent microscope. The number of the yellowish green that represent the micronucleus has been compared to the control group. If the number is significantly decreased from the control group, we determine the agent that we test work as a chemoprevention. This is the study done by Dr. Piengchai Kupradinan and her colleague. She is here. And this is the list of the Thai vegetable and herbs that they study. The neem flowers, Asiatica penniworth leaves, Samnis cassia leaf, Thai bittergourd fruit, and tree product from the noni, such as the noni leaves, Thai noni juice, and noni fruit powder. The ivy gourd leaves, the sesame flowers is called as dog care, and the lemongrass, Chinese kale, and acanthus, Ibratus, called as ngựa and the kaffir leaf. The results show that all of the vegetable except Chinese kale and kaffir leaf show the inhibitory effects to the micronucleus formation in both the direct acting carcinogen mitomycin C and indirect acting carcinogen cyclophosphamide and DMBA. And we would like to know about the effects of the cooking process of Thai vegetable on the micronucleus formation, because in the daily intake, many cooking process have been used in our daily diet. This is the collaborative study between our Institute of Nutrition and the National Cancer Institute. I will give you an example of a kind of vegetable called as Amaranthus species. There are two types of Amaranthus species. The first one is Amaranthus varieties called as amaranth, and the second one is Amaranthus tricolor called as a Chinese spinach. There are three types of cooking process. The first one is raw called as is unprocessed, and the process sample is the blanch and boil. This is the typical use for the daily diet. And the results, mice were given 100 and 200 milligram per kilogram body weight vegetable extract, and after mice received the carcinogen vitamin, we determine the micronucleus formation. The results show that the raw varieties and tricolor show the highest percent inhibition to both standard direct and indirect acting carcinogen. So for the cooking process, blanch and boil have the less percent inhibition to the standard carcinogen when comparing with the raw one. Another example is Gracilaria. Gracilaria is a kind of a red seaweed that naturally grown in the shallow mud lift in the sea of the southern part of Thailand. I show you this picture. It looks like the lady's hairs. We call as Sai Pom Nang or Sarai Pom Nang in Thai. It looks like a lady hairs. And normally for the southern people, they eat them in two forms. It's a raw one and it's called as an unprocessed sample. And the processed sample is washed in the water to become whitening, and is a main ingredient in the salad, in the southern style salad, and for the fresh one, they consume it in form of the side dish with the chili paste. Mice were given diet that contained 1% and 2% of this seaweed. The results showed that the unprocessed Gracilaria had a higher capacity to inhibit the carcinogenic effects than the processed Gracilaria. However, both groups showed the same significant inhibition effects on the standard carcinogens. That's all I would like to say about the short-term assay. Let's move to the medium-term bioassay. Dr. Danai Tiwawet from the National Cancer Institute studied about the 3 kinds of herbs and spice, the pendulata, galangal, and citrus history. The results show the citrus leaf significantly enhanced the pre-neoplastic liver cell foci, while the galangal in both pendulata roots and galangal rhizome had a borderline effect. This is the precaution to use the long-term use of some kind of herb and spice, because they have some effects to enhance the pre-neoplastic liver cell. And for the long-term test, the long-term test has been used for confirm about the chronic exposure of the vegetable and herbs. Miss Anong Thepsuwan and Dr. Pienchai, Dr. Mani Kusambrat studied about the various kinds of vegetable and medicinal plants, such as neem flowers, Thai bitter gourd fruits, sweet basil leaves, Siamese cassia leaf, Chinese kale, Chinese bitter gourd fruit, and acanthus, abratitus, and nasutus curse is a Tong Pantang. The results showed that the neem flowers and Thai bitter gourd fruits, Siamese cassia leaf, acanthus, and nasutus can show the inhibitory effect of the mammary gland carcinogenesis. And the additional neem flowers can show the liver carcinogenesis. But surprisingly, that neem flowers, Thai bitter gourd fruits, and nasutus can induce the neoplasm in colon. I would like to conclude about the study of chemoprevention of some Thai vegetable. The numerous Thai herbs and vegetables contained in our daily food have chemopreventive effects to cancer. It has been suggested that cancer risk can be reduced by eating a variety of herbs and vegetables in a meal. The cooking process, such as blanching and boiling, can reduce the chemopreventive effects of some Thai plants in comparison to the uncooked plant, but they also show the chemopreventive effect to both direct and indirect acting carcinogens. And the chemopreventive activity of Thai herbs and vegetables should be confirmed in vivo long-term assays. And some plants can inhibit carcinogenesis for specific organs, but they have been reported to induce neoplasm in other organs. So, taking a variety of herbs and vegetables should be suggested for health benefit. Lastly, I would like to acknowledge our colleague, our researcher team from the National Cancer Institute, Dr. Pienchai, Dr. Mani, Miss Anong, and Dr. Danai. And I also thank my student Patchayam Masambun and Vilawan Kangwan. And lastly, it's special thanks should be addressed to our animal study in the chemopreventive study. Thank you for your attention.
Thank you very much. So now we have around 10 minutes for question and answer. So I think we would like to invite again the speakers to onto the stage, and if any listeners have any questions, so please give the questions or comments for this session. So please. Any questions? Not yet? So, please.
Thank you very much for your talks. I think all three talks are very interesting and is highly valuable for Thai food research. So, should I appreciate it? Or I think Thai people should appreciate your research. Actually, after the lunch, I was so sleepy, especially 11 hours difference from Bangkok and Boston. But when you start this talk, I completely wake up. And then I could concentrate 3 talks because I feel home. Because it's my area. But even though antioxidant and bioactive food components are not major, but I'm so interesting and a very nice talk. May I ask you some question for the first speaker, Dr. Chulalongkorn? I don't know if I'm correct or not. You found that there's certain Thai food soup, the mixed vegetable soup, reduced the ACF number induced by AUG treatment. Did you ever find that actually that this soup reduced the tumor itself to decrease it? Because ACF is a precursor of cancer, but sometimes not all ACF goes to tumor. So I wonder if you really checked this soup really reduced the number of tumor. And then the other one is, did you see that ACF from the small intestine or large intestine? Because the chemical calcium would frequently more develop ACF in the small intestine and more developed in the tumor in the small intestine.
Thank you, Professor Shaw. The first question, as this is the thesis of a master student, and I think it's I'm quite sorry that it's not too long to cut the tumor as you asked, because the period of study is just 6 weeks. It's not close to the medium term, just in between short term and the medium term. And what we investigate is just the ACF and also the detoxifying enzyme. That's enough. And for the second questions,
so did you find the ACF in the small intestine or colon?
In the colon. Yes.
Colon could be more valuable because human doesn't have small intestine.
Yeah, exactly.
Yeah, thank you. So how many times did you inject the ACF? Once or
2 times.
2 times.
Yes.
And then sacrifice 6 months.
Yeah.
So you can find the tumor because tumor.
Not only 6 weeks, not 6 months.
6 weeks.
Yeah.
So you usually found the tumor 20
Yeah.
weeks later. So you cannot find the tumor, but see the ACF.
Yeah.
Okay. How could you give the vegetable soup to the animal? Using the tube or extract?
Gawashing. We cook as usual, that we age and then we freeze dry it, and then we resolubilize as I calculate as if we assess 60 or 50 kilogram per people, we eat 1 cup of soup, and equivalent to the weight of the rat. And then we feed them, one group is 1 serving and the other group is 2 serving, just by the watching. Yeah.
Is anyone have a question? Can I continue to the second speaker? Yeah. Dr.
Me like this. This is for Professor Choi only.
No, I'm sorry, because if anybody has a question, go ahead.
Yes.
Actually, if I continue, then I will be more than an hour.
And because it's good for others as well.
The second speaker, Dr. Tunti Popia.
Yeah.
The inflammation and cancer, nutrition and inflammation and cancer is the hottest topic in the current year. And then everybody in our research center study on the inflammation and cancer. It's a very important subject, and I think you've demonstrated your Thai herbs and the spice one that can reduce the inflammation. Yeah, that will be great studies. I just wonder what kind of mechanism do you think by which these Thai spice and herbs convey the anti-inflammatory effect?
Actually, we process for our current study. We investigate and already know that it blocks the signal pathway, the MAP kinase, and also inhibit the activation of the NF-kappa B. But I didn't demonstrate because I thought that the audience
demonstrated. You would do it.
Yeah. But I know that the audience is not signed, so I didn't raise the result. Okay. But we studied in the mechanism that it's blocked the MAP kinase, the ERK and JAK and P38 phosphorylation, and also inhibit the activation of the phosphorylation of the NF-kappa B, which is the transcription factors of this pro-inflammatory gene.
Right. I don't know if you are familiar with Dr. Roderick Dash, who is good at the nutrients and inflammation. And then I show you I give you my book to recently published Nutrients and Epigenetics. He wrote a chapter there, how bioactive food component reduce the inflammation. The theory is this, and can I talk? Yeah. The thing is this, you show that sulforaphane from the broccoli, the isocyanate, and the garlic component DDS, diallyl disulfate, and butyrate, short-chain fatty acid. Those bioactive food components are histone deacetylase. Okay, this is one thing. And the resveratrol and curcumin, the curry component, which has a lot of function. Both of resveratrol, they are from the grape skin, and those are the curcumin. Also has histone deacetylate activity, and thereby decrease the inflammation. You mentioned that NF-kappa B activation. Then once NF-kappa B is activated, what happens is it acetylate P16, and thereby acetylate all the pro-inflammatory downstream gene. So that histone acetylations most of the time if acetylate the histone like H3K9 in the DNA, they activate the gene. So that is make the pro-inflammatory and inflammation. On the other hand, if you use corticosteroid or theophylline, or resveratrol or curcumin, because it's histone deacetylate. So deacetylate all the acetylated genes by NF-καπνίδι. So that we depress gene expression. So all the pro-inflammatory genes are depressed. So inflammation is depressed. So because from your certain chart, I saw that epigenetic modification. So histone acetylation is one kind of histone modification that directly affects the gene expression. So I suggested to study about this way. I mean that such a bioactive food component in the Thai spice or anything. I don't know. We cannot explain everything, but some part could convey by histone acetylation, so-called histone and epigenetic phenomenon. Thank you too much. I'm sorry. Oh, the third speaker. I saw it was so interesting that Dr. Butri. Yeah, I don't know. So sorry. It's a very nice study, but because I don't know it, so I'm asking you this question. You check the